All right, and here we go with learning and memory part two. For those of you that are just coming from part one, I would definitely suggest taking a quick break here. This material will go a little bit long. And for those of you that have not yet looked at part one, you're going to need to before you start this lecture. The material here is going to build off of some ideas presented there. And without that background, you're going to have a hard time.
In talking about learning and memory, one of the things that we need to focus on first is almost the opposite of this in talking about amnesia, because we're going to be talking about parts of the brain that are involved in specific kinds of memory, and it turns out that these are different. So anterograde amnesia is amnesia that occurs after a head injury, meaning that you have trouble with remembering things going forward. And if you remember anterograde tracing, this is tracing from an origin onward or forward. And so this matches with those definitions as well. And then retrograde amnesia is the one that you probably most often see in the movies, where somebody hits their head or has some sort of a brain damage or traumatic event, and they have trouble remembering anything that's happened in their past. So this is the one that Hollywood really likes to play on because people forget what they're doing. And I can think of a couple of examples. One of these is Going Overboard, and another one is 50 First Dates. But since I and everyone else is spending a lot of extra time in their house, and I think we're all looking for something new to watch on Netflix or Amazon Prime or Hulu, if you get to this slide, pause and take a minute. And if you can think of a movie where you remember that they've used the concept of amnesia, put it in the classroom chat, and then everyone can go look and see what movies have something about this form of memory. So as we've been talking about memory, we've talked a lot about patient HM. And patient HM, if you remember, had very severe seizures, and he went in for a procedure that was pretty much a lobotomy, and it caused some damage that was supposed to eliminate the seizures. And in fact, that is what happened, but it also caused him to have severe anterograde amnesia, so a problem remembering things that happened after this brain damage. And this led to a couple of really important observations about the role of the hippocampus in learning and memory. The first thing that was at least predicted was that the hippocampus must not be the place where these long-term memories are stored, because HM could remember all of what had happened earlier in his life. He could remember where he grew up and where he was born and who his parents were, and all of these things had already been stored, and he could remember them, which also says that he is able to retrieve these memories. So there were no problems with either retrieving or remembering these long-term memories. There is another thing that is a little bit less obvious, and that's that the hippocampus must not be the location where short-term memories are stored. And this is a really important point because patient HM was able to carry on conversations for periods of time. He was almost able to do everything in his life normally, except for the fact that anything that happened after his surgery, after some very, very short window of time, he would not be able to remember that anymore. So in the moment, these very, very short-term memories were still intact. So what this suggested, and this is kind of the primary hypothesis that came out of patient HM symptoms or Henry Molaison symptoms, is that the hippocampus must be involved in converting short-term memories to long-term memories, okay, in converting them. So what this means is that patient HM could interact with you normally, and he could carry on conversations, he could do everything for a few minutes, but then when it came time to take that short-term memory and turn it into a permanent memory, there was a problem in the consolidation or the storage of that memory for long-term use. So you may not be surprised to learn that patient HM is not the only patient that has ever presented with these type of symptoms. And another famous patient, although not as famous as HM, who was institutionalized for much of his adult life and able to be the subject of study while he was institutionalized, is a patient that's now known as patient RB, who had brain damage due to heart failure, and he had what you would call hypoxia, so a lack of oxygen to the brain, and had the same exact symptoms as patient HM. He wasn't able to form new memories after this damage. And it turns out that he donated his brain to science, and so after his death, they figured out that not only was there damage to the hippocampus, but to the CA1 region of the hippocampus, this very small subregion here. But it turns out there's something very, very important about this subregion, which is that it's rich in NMDA receptors, those receptors that we talked about that are kind of critical for causing a synapse to become stronger, to strengthen. We're going to talk about that in section 4 here, but keep that in mind as you go through this. So what we now know is that somewhere between 1 and 30 days after a memory has been formed, it transfers outside the hippocampus. And that during that 1 to 30-day period, the hippocampus is doing something like a replay of these memories. And this is really important for taking those short-term memories and converting them into long-term memories. And so people like patient HM and RB aren't able to do this because they don't have that sort of medium-term memory that's happening there. So if an individual has damage before hippocampus damage, they retain these. But in this critical period where the memory is in this replay mode, then the memory can really easily be disrupted, especially from you can especially have disruption from consolidating that memory. So what all of this comes together to tell us is that the hippocampus is really important for the retrieval of memories for a critical window of time that lasts about 30 days. And this is why I jokingly say, but also with a slight element of seriousness here, that maybe we should be considering a middle term memory, because there is clearly a window of time where you need the hippocampus, and it's not necessarily a short term memory anymore, but it hasn't been converted to a long term memory. One of the things that we see too that supports this is that in individuals like patient RB or HM that have had some kind of damage to their brain, that they get this really, really short window of retrograde amnesia. And this is not like when somebody wakes up from a surgery and they can't remember a few minutes before they went to sleep, which sometimes happens. These individuals have lost an entire month of their life in some cases before they have damage to the hippocampus. And this is consistent with the idea that there were some elements of their past that were stored in this middle term memory, and only those ones were disrupted in terms of long-term storage. And another thing that we've seen is if you look in human imaging studies that memory that's been there for a really long time doesn't cause much activation of the hippocampus. But if you ask somebody to recall a memory that they just formed a day or 2 days ago, that you get really strong activation of the hippocampus. And so it turns out if you record this individual's brain every day and ask about that same memory as a part of the study, then what you're going to see is the activity of the hippocampus from day 1 through around day 30 is going to go down and down and down. So one of the things that I've been saying is that we should consider the brain as a series of separate systems. And if you remember, one of these systems we talked about is involved in stimulus response learning. And so something that's kind of interesting is that individuals with hippocampus damage are still able to perform these stimulus response type of tasks very well. And one task that's been used for this kind of repeatedly is called eye blink conditioning, where an individual is going to have a tone played in their ear, and then they have a little device that can give a puff to the eye. And as all of you know, that if you get a big old puff of air to the face because somebody blows in your face or there's a big gust of wind or something like that, then you're going to blink. And so eventually this response can become conditioned. So what it turns out is that individuals with damage to the hippocampus are able to show this kind of learning. So they can retain this response, and if you train them on this task, they get better and better at it over time. And then if you test them a month or 2 months or 3 months later, that those changes in their ability to correctly associate the tone with the eye blink, or their ability to have a stimulus response memory, is maintained. And so what this suggests is that from data from both human and animal studies is that the stimulus response learning does not require the hippocampus in order to happen. It's a separate memory system altogether. It also turns out that patients with damage to the hippocampus have no issues learning motor responses and remembering these over a period of time. So if we look at motor learning, or what forms after that, motor memory, patients with anterograde amnesia from hippocampus damage don't have any problem with that. The way that the serial reaction time task works is that there's an asterisk that would appear over one letter. You all probably can't see that. Let me try with a different color. Over one letter on a screen, and the patient is expected, or the person that's being studied, the participant, is expected to make the response over that asterisk. And they're going to do this in a long series of kind of random nonsense letters. And so the measure that comes out of this, as you might guess, is reaction time. Now, the thing is that there's a bit of a trick the experimenters play here, because you could sit there and you could say, well, okay, I can just get faster at pressing keys because I can get better at pressing, DCBA. But it turns out that the order is given over a few days, and it's not random at all. The order is actually becomes predictable, and so people can get better at making this exact motor pattern, even though they're quite unaware in most cases that the pattern is repeating. They just get better at it because the sequences that their fingers go through get better, right? There's a very specific set of synapses that are being activated here. And so individuals with damage to the hippocampus have no problem becoming better at this task. And they will learn the task just as well as anyone that's a control that doesn't have damage. And there's a lot of anecdotes here that also support this, where if you ask an individual that has damage to the hippocampus to learn something new, which for patient HM might have seemed like the kind of thing that would be tricky, but actually he was studied in many of these different kind of tasks. And so if you ask somebody to learn something and get better at it that is completely motor memory, like knitting or playing a video game or tracing the same pattern over and over again, they can do that just as well too. So there is an increase in skill and a demonstration of some sort of knowledge of what they're doing, or at least a memory for how to do this based off of the fact that they get better. So the third type of learning that we need to talk about is perceptual learning. And remember, this is something where we get better at our ability to recognize stimuli and identify them and all kinds of things like this. And so one of the ways that this ends up being tested is that individuals or participants or patients, whatever way that you want to look at it, will be given a series of drawings. And what's going to happen is that the drawing is going to look really incomplete. And then the experimenter is going to present the next one in the series that'll look a little bit more complete. See if you can start to kind of guess what these are. And usually by the third one, people can start to make out what this is. By the fourth one, almost everyone should have made out what it is. And by the fifth one, you should be able to see that we have an elephant and an umbrella here. So not only do people get better at this depending on the number of pictures, but they usually get better over just a couple of sessions of doing this. So this exact type of experiment was actually run on patient HM, and he was able to improve at the perceptual learning. And he did demonstrate also long-term retention of this task. So he got better at it and was able to maintain how much better he was over a window of time. But there was one thing about this where he was never quite as good as an individual that didn't have damage to the hippocampus. So what this suggested, at least, is that some aspects of this task might rely on the hippocampus, even though you also can't say that a patient with hippocampus damage cannot show perceptual learning. And as it turns out, in other studies since then, there's been a suggestion that this may even depend on the type of perceptual learning task that's given. In fact, that there's some of them, as hard as we try, that are not purely perceptual learning tasks, where individuals who are maybe using different strategies, like an individual that doesn't have damage to the hippocampus might start to use something like a spatial or a relational strategy to solve this, which would explain why there might be a problem. And this brings us to the last type of learning that we need to talk about, which is relational learning. So for 3 different types of learning so far, we've said that patients with hippocampus damage are able to perform these types of tasks, and they're able to improve at them. So they're showing some evidence of learning. However, if a patient has hippocampus damage, then they will have all kinds of problems with tasks related to relational learning. So what this tells us is something even more than the hypothesis we started with at the beginning of this series. We said that the hippocampus is responsible for converting short-term memories into long-term memories. But we have to make that even more specific. What we have to say is that the hippocampus is really important for converting short-term declarative memories into long-term declarative memories. And this becomes much more specific because anything that falls into this procedural category, which you can go back a section and see everything that's there, but something like riding a bike, as you see here, is a great example. Those things are fine. Another thing that we see in individuals that have had the kind of damage to the hippocampus that patient HM had is that they have trouble with spatial memory. And we already talked about this when we just defined relational learning, that spatial memory is a type of relational learning. You have to learn this relationship between all kinds of arbitrary cues in your environment. Turn left at the McDonald's and turn right at the church, and those kind of things get us through our world. And so it shouldn't be surprising that these patients would have trouble. And one of the things that verifies this is that there was actually a really famous patient called patient TT who was a taxi driver in London for a really, really long amount of time, years and years and years. And so this taxi driver had an incredible mental map of London. And it turns out that if you asked him where to go, he could tell you because he already had a long-term memory for all of the different streets and turns, and he could give you directions, better than most GPSs could. And that's saying something because that's an incredible spatial memory that he has. However, in trying to navigate new places or learn new sets of directions, even he still had trouble. So someone who had a very, very strong spatial memory wasn't able to form these new spatial memories after that damage. Of course, one of the last things that we haven't mentioned yet is that there have been thousands upon thousands of animal studies now showing that if you have damage to the hippocampus, that you have trouble being able to form spatial memories. And we saw the example of the Morris water maze in one of the past two sections, showing exactly that if you have a lesion of the hippocampus, or if you have a temporary inactivation of the hippocampus, that animals will have trouble forming these relational learning memories. They'll have trouble with spatial learning, among other things. And something that's a little bit more correlative, but makes a lot of sense based on what we said, is that in humans, if people do a structural fMRI and they look at the size of the hippocampus in humans, or also if you take the hippocampus out of animals and you look at the size of hippocampus, that animals that have performed much more spatial tasks, or humans that have a better spatial memory or have a better sense of navigation, tend to have a larger hippocampus. And this goes along with everything we've already talked about for sensory systems, where the size of a brain region or the amount of neurons and axons devoted to something tends to relate back to the resolution of that system. And so if you happen to be blessed with the slightly bigger hippocampus, you're going to do better in spatial learning. And so here is that Morris water maze idea that we talked about. So just as a reminder, they're kind of two different strategies that somebody could use to solve this Morris water maze. If they start in the same spot every time, they can learn a strategy where they just learn by direction to go diagonal and to the left in the case of this mouse. But in a task that forces relational learning, where there's 4 different start positions, then individuals have to learn something about the cues in the room, like where this door is, because there are at least, for these starting zones, 4 different paths that you would have to take. Well, it turns out that there was a study by Iaria and colleagues around 2003, and what they found is that there are some individuals that might use either strategy, and that this is by choice. So what they did was they put a lot of participants into a virtual reality maze, and then they asked them to solve it. And some individuals make a fairly long series of stimulus response learning decisions, where they turn the stimulus response learning into a chain, and they will just, instead of learning the cues in the maze, they just say, okay, I go left, right, left, left, right, left, right, left, left, right. And that can fall into a category of stimulus response learning. Whereas other individuals will look and they'll notice the cues on the wall. I turn, left at this painting and right at the stripes. And they use all of the different spatial cues and they make a mental map of what's going on in the environment around them. And this doesn't quite break down 50-50, but there are enough people that use each strategy to where they were able to study them. And what they find is that, sure enough, individuals that use the relational strategy, they have far greater activity in the hippocampus, right? Far greater blood flow from this fMRI imaging. And others that use the stimulus response strategy have more activation of the caudate nucleus, the stimulus response memory system. So this validates everything that we've been saying, that there are different systems for different kinds of learning and memory, and that depending on which one you're engaging, you might actually have a very different way in which the memory is formed and stored, and even your ability to recall these memories or to demonstrate that you've had this learning and that you have this kind of memory might be different. Well, you've almost made it out of learning and memory, but unfortunately, I'm just going to let you know this last part is kind of the meat and potatoes. It's the beast of all of this, and it's really the one part that if you walked out understanding anything about learning and memory, that would be the most important. So we already talked a little bit about the Hebb rule, and I've actually kind of laid it out here a little bit more clearly for all of you. Just in case, as I'm explaining it in the past lecture when we're just talking about stimulus response learning and classical conditioning, that it doesn't make sense. So what we have happening here is that we have some neuron of a weak synapse, like this 1,000 hertz tone, that repeatedly becomes active about the same time as this blink neuron fires, right? And the reason that the blink neuron is firing is because we have a puff of air to the eye that's driving it. And what this seems to do is it seems to somehow change the structure or chemistry of this synapse here. And so you have to remember that the synapse is going to have two parts. Of course, we're going to have the part coming from this purple neuron, and that part is going to be the one that has a bunch of neurotransmitter to release, and it's going to be doing some signaling, and that's why we say it could be a chemical process. And then we also have the receiving end of this synapse. And this receiving end is the one that's going to have various receptors to get the message. And so the idea is that over this repeated activation, that what we have is some sort of a strengthening of the synapse so that the excitatory postsynaptic potential, right, which really just means the amount of depolarization that's happening here, or the number of votes that that this potential, this dendrite is bringing into the neuron to tell it, to fire an action potential or not, have become greater. It just becomes a stronger message that's coming through at the most basic level. Okay. So one of the questions that, of course, we needed to ask if Hebb's hypothesis were to be true is how would something like this be accomplished? Well, one way that it can be accomplished is by the timing that we have here. One important element of this seems to be the timing, that we have some very strong stimulus, that a puff to the eye causes a blink. These two are very close in kind of the relationship and the response. We have a sensory stimulus coming into the eye, and we have a motor reflex in the form of the blink that the eye is making. And so the idea is that somehow that activating this strong synapse at the same time as this weak synapse can cause the weak synapse to become stronger. And there's another prediction here that goes along with the way that learning happens, which is that repeated pairings should make this incrementally stronger, so that eventually we don't even need the air puff to the eye. And as we just talked about for some of the different learning tasks, even one for stimulus response learning, the eye blink conditioning that many people with all different kinds of brain damage, including patient HM, have gone through. And it turns out that they can learn these things just fine in certain systems. So that shows that they're having this sort of what you would call, maybe an incremental improvement over time in um, a stimulus response task. So a lot of work about how memories become formed and strengthened and how synapses become strengthened has been done in the hippocampus, kind of for obvious reasons, because for a long time people thought this was the seat of all kinds of memory. And a set of experiments that have been done involve the primary input to the hippocampus, this entorhinal cortex, and it actually travels or has a bundle of axons that travel in this pathway called the perforant path. And they're going to make synapses in a part of the hippocampus called the dentate gyrus, which is pretty much the primary input of the hippocampus. This information then travels through CA3, the CA3 area of hippocampus, to the CA1 area of hippocampus, and then this can kind of go out anywhere to the brain. And so it turns out if you electrically stimulate this perforant path over and over again and just generally activate the hippocampus, you can see an increase in synaptic strength, and it can last for months. And this is the big picture overview of that shows us that some sort of strengthening of synapses is happening. So the stimulation being that it's of the perforant path, one of the things that this tells us is that really by stimulating the axons coming through and not at the cell bodies or other things, that it's most likely that whatever is happening to strengthen this synapse is happening at the synapse itself, at the connection between these two neurons. And it's not something that's happening back at these cell bodies because we've kind of cut them out of the picture here. And another thing that's been shown here is that this needs to be repeated for the synapse to strengthen. So you can't just stimulate one time. If you stimulate one time, you don't really see much of an effect. And so you can see this actually in this diagram here. If you look at a single stimulation of that perforant path before long-term potentiation, then you do get an excitatory postsynaptic potential. But what happens after you stimulate repeatedly is that this excitatory postsynaptic potential, not the size of the action potential, because remember our all or none rule, the action potential either fires or not, and it stays the same size, but the postsynaptic potential, right, the amount of current, the amount of ions coming in to this synapse, which is really the reference I always go back to is that this is the number of votes that have been given to this neuron to decide whether or not to fire an action potential. So now we have more votes. We have a stronger connection that's giving more influence that says, hey, you should fire an action potential, this excitatory postsynaptic potential. And there's another interesting thing that happens here, which is that at 24 hours later, the synapse will go back to normal if you give a single pulse. And you look at kind of the population activity here. But after 2 days, we see this recovery and then kind of the long-term maintenance, even out to a few days, where the excitatory postsynaptic potential becomes strengthened at this kind of like 48-hour time point and stays strengthened for quite a bit of time. So what this suggests is that there are a couple of different components to this. There's something that happens immediately when you've repeatedly stimulated a pathway that we can see in about an hour after, and that there's some way that this gets converted into something that's more long-term on the order of these 48 or 96-hour time points that we've looked at in the other traces here. Another thing that seems to be important for this is that the stimulation has to be rapid. So if you take these pulses and you stimulate, let's say you stimulate 100 pulses and you do that over 100 minutes, one pulse a minute, which is very, very slow, probably slower than most neurons would naturally fire their action potentials. It won't cause long-term potentiation. But if we make the stimulation very rapid and we make it look like a big input coming in all at once, if you stimulate 100 pulses within 1 minute, as if there's a really strong signal coming in from some other neuron, then it's much easier to produce this long-term potentiation. So it seems to rely on the presynaptic cells, the ones that are sending out the signals, to have something that is kind of a strong signal coming in the. So what we think happens, and this part might be a little bit difficult to get, and it's probably even a little bit difficult, I'm going to be honest with you, in this e-learning format. So bear with me here, and for this section, if you kind of drifted off a little bit, come back and pay attention. So what we think happens is that if we have a neuron that's already depolarized, and so remember, depolarized means that it's kind of primed to fire an action potential, then what seems to happen is that if we stimulate one axon that's coming in, or really one synapse, and only one synapse, if you do this in an incredibly controlled setting, then what ends up happening is that only that synapse that was activated while the cell was depolarized is the one that's strengthened. And if you stimulate this synapse independently, you can do it quite a bit. But if you stimulate it independently and we take away the fact that we've depolarized this cell at the same time, but we just stimulate, then all of a sudden the synapse is no longer strengthened. So what this suggests is that something really important happens when this cell is depolarized. So the timing of the spike dependency matters. And if you think about the example where we have kind of neuron A for a puff of air coming in, and neuron B that's a tone, and then neuron C that they're both communicating with in this eye blink example we've gone back to a few times. What this suggests is that it's really important that this neuron C is already been activated, right? It's already depolarized and ready to fire an action potential when neuron B sends a signal, in order for just this synapse to become stronger, stronger, stronger. So why is that? Well, this all goes back to the idea that we introduced very, very briefly a couple of sections ago on the NMDA receptor, where these NMDA receptors are receptors that bind the neurotransmitter glutamate. And what they have is this big magnesium ion that blocks them, so that ions aren't able to flow in and out of the cell. So it turns out that what happens if the inside of the cell is depolarized is that it will actually push this magnesium ion out of the way and make it to where it can no longer do its blocking, and this allows calcium to come into the cell. And when we talked about chapter 2, I kind of said, hey, forget about calcium because it has some tricks that it plays, but mostly it's involved in making changes to the cell. And so it turns out that for these NMDA receptors, then they allow calcium often to come in as one of the things that they'll allow through. And this can then allow all kinds of changes to happen right at this synapse where the calcium is coming in. So when calcium comes into the synapse, there are a few things that can happen. The first that we've already talked about a little bit more is that second messenger systems can be activated. So these are these metabotropic receptors where you have to have kind of a cascade of things happening. So calcium starts to activate those, and they can remember whatever effects happen on these metabotropic receptors can happen for a long period of time. So the neuron can kind of stay depolarized for a long period of time just by having calcium come in. That might explain why after an hour you still see a long-term potentiation. We can also activate all kinds of different enzymes that these various enzymes can have specific effects on what's going on in the cell, and they can start to cause more production of proteins or less production. They can start to disintegrate things. So you can activate other sort of chemical processes in the cell. You can start to trigger chemical reactions or reactions between different organelles within the cell. And one of the things that we've seen as a result of this, all of these things happening, is that you can get an increase in the size of a dendritic spine, which actually relates back to a change in the conductance of the dendritic spine. So what this means is that the dendritic spine gets bigger, and if you remember everything we talked about in chapter 2, one of the really, really important principles that I asked everyone to walk away with is this idea that the surface area that gets activated is another way of sort of changing the number of votes, the amount of influence that a certain part of the cell has. And so this means that if we have a synapse that happens at the cell body where there's a whole lot of surface area, it gets many, many more votes. It can cause a greater change in current or conductance than a part of the cell that's small, like a dendrite that's way off at the end of the cell and is teeny, teeny, tiny in diameter. And one of the things that goes along with all of these changes as well that's equally important of activating enzymes and causing all of these changes within the cell is we know that we can get an increase in AMPA receptors. So remember from our introduction, these AMPA receptors are related a little bit to the NMDA receptors in that they're another type of glutamatergic ion channel, right? They bind glutamate and they allow ions to come in. And the two of them tend to kind of co-occur at the same synapses. So what seems to be the case is that if we put all of these things together, we need calcium to enter the cell in order to especially cause changes in the dendritic spine and AMPA receptor. And the way that we can do this is to have this long-term potentiation happen. The way that this happens is to make that synapse more excitable in the future, and a primary way that you can do that, as we hinted at before, is to increase the number of AMPA receptors, because these don't have that big magnesium block in the way that the NMDA receptors have, and so it takes a lot to move that out of the way normally. But if you have AMPA receptors that are unblocked, then ions can just pass through when the neuron fires an action potential. And a stronger synapse is going to look something like that. Pardon the terrible drawings here. And what seems to be key for this, which makes sense with everything we've seen about how we learn things, how we pair things together, is that we need for the neuron in which we want to induce this long-term potentiation, this long-term change in the synapse, this long-term change in the ability or the potential for this synapse to cause the neuron to fire an action potential, is that we already need an action potential happening. So this is, in a way, how we pair two events. If the neuron's already firing an action potential and it happens to get another signal, then it becomes more likely to listen to that signal in the future. That signal that comes in while it's already being activated starts to carry more weight, to have more influence. So what we think happens to kind of put all of this together is that when we have calcium influx, that one of the things that this calcium influx causes is an increase in these AMPA receptors. And there's a mechanism for this that has been figured out that makes a lot of sense, which is that if you add more AMPA receptors, then it becomes easier in the future for these NMDA receptors to become unblocked, which means it becomes easier for calcium to enter into the neuron, and then it becomes easier for this dendritic spine to become bigger and stronger in the future. And what we think happens to add these AMPA receptors on is that AMPA receptors, for people who have used a lot of different fluorescent markers for the AMPA receptors and looked at these under microscopes that are very, very high powered, is that there are AMPA receptors that can be on the dendrite, but not really on the active portion of the dendrite. So they're kind of put away for storage. And what seems to happen is that when you have all the calcium enter the cell, that some of these activate enzymes which can cause the AMPA receptors to start to move into the spine and to eventually come in and populate the important part of the spine where all the neurotransmitters coming in and binding to send new messages. And so then this means that the cell is going to recognize that it can store a few more. It's going to bring some here to be able to be stored. And then there's another set in the queue to where we can keep adding more and more and more AMPA receptors onto the cell, depending on how strong we need this synapse to be. So there's a pipeline for making these synapses stronger over time. So, and I guess what I'm saying here is I'm trying to kind of catch my thought is that if we want if we want this long-term potentiation to last, then this requires that we start to produce more receptors, that the receptors get moved into place. So there's kind of an entire pipeline starting from the cell body, getting signaled that maybe we need more AMPA receptors, all the way up to putting more AMPA receptors on the synapse that leads to this change. And a lot of that depends on having a synapse that was activated in the first place, where we were able to cause this change by getting some calcium into the cell to trigger this cascade. So there's a kind of microscopy that we didn't really talk about much when we went over methods called two-photon microscopy. And in this sort of setup, there are some experiments where people can actually conduct them in and look at living neurons. And so that's what's happening here. And they can also, in some cases, use the microscope to stimulate and cause a synapse to be activated and monitor it over time. So what happens is if people induce long-term potentiation by repeatedly stimulating, and then they can also take images of what this dendritic spine looks like, you can see that over long-term potentiation that a synapse can form if there's an input that's coming in, and it can actually even get bigger and stronger and have more surface area after extended periods of time following the stimulation. So we can actually see these changes happening, which is why we know that they can truly happen. And also, even though I'm not showing it here, some of the changes that we know to happen that we've also observed using either the confocal microscope, which we did talk about, or this two-photon, which is even better resolution, are those changes in the number of AMPA receptors that are on the dendrite. Another thing that's been coming out more recently is that long-term potentiation might not just be unique to the postsynaptic cell. So to this point, we've been saying that there is some sort of dendrite over here that's receiving this message, and that what we're doing is we're increasing the number of AMPA receptors that are on this synapse in order to make it more likely that it's activated. But another way that you can change what's going on and how easy it is to activate this neuron over here is that you could actually come in very simply and change the amount of neurotransmitter that's being released from low to high. And so it turns out that we think this might be happening too. So if you cause calcium to enter into the presynaptic cell, you can cause quite a few changes. And there are even some messengers that can come from the postsynaptic cell back to the presynaptic cell to activate the presynaptic terminal and cause changes. And one of these that happens to activate and allow calcium to come in is called nitric oxide synthase. And so there's some growing evidence right now that this nitric oxide synthase might be released and act as a retrograde signal, so going against the direction of kind of the message between neurons. You know, the message is going this way, and then the nitric oxide synthase comes back the other way and tells the presynaptic synapse, right? This presynaptic synapse to release more neurotransmitter. So that is yet another way that we think all of this processing might be happening, is that you can simply have a change from a low amount of neurotransmitter to a much higher level of neurotransmitter being released. And that means that, again, the postsynaptic cell is getting a stronger message, more likely to fire an action potential, and more likely to be listening to whatever this synapse might be.
And just as a bit of a fun way of closing, what I want to encourage all of you to do is to go check out the long-term potentiation video on 2-Minute Neuroscience. I'm a big fan of sending you all over to different videos when I can, especially if the way that they present things might help to summarize things or might connect better with your learning style. And in this case, the 2-Minute Neuroscience video on long-term potentiation does a great job of just taking everything that I've done over the past maybe 10 or 15 minutes and condensing it down into 2. And they have some really incredible animations that will help you kind of understand everything and put it all together. So I'm going to point you to that direction and highly encourage that you all go take a look at that video. It's worth your time to watch it. It probably will help you connect some of these ideas together. And with that, I hope everyone has a great rest of the day and rest of the week.